forward to the corner. Okay, you should see the
There
recording.
you go.
Great.
Okay.
Now I can stop sharing. Now you can begin sharing and we can officially take it away. So go ahead.
Okay, thank you again, John, and thanks to AWA for inviting me. I've always been a fan of your organization for years and look at it as sort of a model in many ways for how we'd like to see monitoring programs evolve and develop. That is, having more centralization in different regions, and in some cases, even developing satellite laboratories. But we can talk about that at a little bit later, perhaps, if anyone's interested. I want to thank everyone for coming out tonight and giving up the evening news, perhaps, although and being willing to listen and talk about our lakes and cyanobacteria in our lakes. It's a topic that I've just can't get enough of, it seems, as we keep developing new ideas and discovering new things. As John mentioned, we used to think that cyanobacteria were just those things which we could see at the surface of the water at certain times of the year. And now we find that it's much more involved and interesting than that. And I'll touch upon some of that tonight. So I'm sure that most of you have encountered the term cyanobacteria. So I'm not going to give a lesson in cyanobacteria by any means. I just want to touch upon some aspects to put them in perspective, and then at least touch upon some of the questions that John had suggested I would be talking about. I should cover most of those in one form or another through the evening. So as he mentioned, I'm at the Center for Freshwater Biology at UNH. We have an ecotoxicology laboratory there. You're all welcome to come down and visit sometime. We are getting our new laboratory sort of as we speak. We'll be moving in about 2 weeks. So it should be interesting to see how what we can do there that we haven't been doing. And I think it's going to be actually a nice improvement over what we have. So feel free to contact me if you'd like to have a look at what we do and how we do some of these measurements. So tonight I am going to talk about sort of cyanobacteria, the origins of them, the risks associated with them, and sort of what we can do besides talk about them. So let's start off by simply addressing the question of what cyanobacteria are and why are they in our lakes. The audacity of cyanobacteria in my lake, of all things. If you put them in perspective, and the perspective actually comes from a very interesting thing, which is most bacteria are, of course, as you know, are very delicate, and they don't preserve well in fossils. So it's hard to find the age of most bacteria, but cyanobacteria, one type in particular, actually creates these lime concretions by having photosynthesis, which raises the pH. And then the lime, actually, the calcium carbonate precipitates out. And over hundreds of years or even longer, they form these large mounds. I've seen them in New Zealand, they're in Australia, they're in several places. You can see them in the oceans. And these mounds actually represent huge colonies of cyanobacteria. And one of the interesting things is that they repeat so that every year they form another layer, so you can actually age the colony. But most importantly, you can identify that this was made by cyanobacteria. The interesting thing is that not too long ago, there were a couple of researchers out of Canada that were looking at evidence of fossils in some of the oldest rock that we have found so far, which is in northern Greenland. And that rock then, they were able to find evidence of the stromatolites in this rock that aged the cyanobacteria back to about 3.7 billion years. putting that in perspective, the best estimates now for the age of the Earth is only 4.7, 4.8 billion years. Seems to be fairly consistent now with newer information coming in. So what it means is these are some of the earliest forms of life that existed. So we sort of imagine the Earth at that time, nothing else alive except maybe some other bacteria and cyanobacteria. And they owned everything. And they adapted to all sorts of habitats. And there was no oxygen at that time. So the Earth was a very different place. But they essentially evolved to those conditions. They polluted the environment with oxygen, which of course made it possible for us to evolve eventually, and then adapted to those conditions as well. So when you look at it, they were they occupied the Earth pretty much as their own for probably at least 1 billion years until the algae evolved. And then they had some competition with them. And they actually appear to have been very important in the development of the evolution of algae as well. So they're old, they've always been here. No surprise that they're here now. The question is, why are they a problem? And what can we do about it? So let's take a look. Well, one of the things about being a bacterium, algae essentially are not bacteria. They produce membranes, and they can produce modes of motility that bacteria don't have. But being a cyanobacteria, the one thing you could do really well was produce chemical compounds. So that's their specialty. And in fact, today, some of the compounds we'll be talking about are unique. They're only produced by cyanobacteria. So that's really quite interesting in itself. It appears that a lot of these compounds were used both in helping them gain nutrition, but also as a means of combating their rivals. That is, their essentially their other bacteria that were wanting to occupy their place. So they were essentially used as ways of a defense against others. So of course, there were no humans around that time. So many of these compounds have been around long before we were around. So here's some examples of the types. There are literally hundreds of different types of chemical compounds that are produced by cyanobacteria. And so let me just talk about a couple of these so that we have them in our vocabulary at least. And they're often lumped into categories of toxins according to how they affect vertebrates. So the hepatotoxins that have the main effect is on the liver, but now we're learning can be other organ systems as well. Microcystins are some of the first that were discovered. In fact, some of the first discoveries were made at UNH before I was part of that organization of that lab. And they discovered that microcystins, when injected into rats, caused rapid death within about 4 to 6 hours. And they would do this by essentially destroying the capillaries in the liver, and then the system would break down. And so they are very powerful toxins. And the others are cylindrospermopsin, which is also a liver, considered to be a liver toxin, and nodularins, which is found probably in Great Bay and some other brackish waters. The neurotoxins are very interesting. The anatoxins, which are very fast acting, and the neosaxitoxins, which are also found in freshwater cyanobacteria, look very much like the saxitoxin in the marine system. And then there's BMAA, which is a relatively more recently discovered toxin that's produced by most cyanobacteria to some extent. And it's the one that we see some linkages to neurological disorders, as well as their direct effect if they're consumed. So I wanted to just show you what we're doing at UNH in terms of just putting it in perspective as what our interests are. We've been working with microcystins since I joined the group in 1987, this lab, and now it's changed obviously in the meantime in terms of the people involved in it, but we're still studying microcystins. It was one of the first discovered, and it's one of the most widespread of the cyanobacteria toxins. We now know that in addition to liver diseases and tumors, it can also affect the brain, it can affect the nervous system, so it has more than one effect, even though it was originally referred to as a liver toxin. BMAA is a really tiny amino acid. And as small as it is, you wouldn't believe how the damage that it can cause. It's one of the ones we're studying because it's so widespread also. It's not very well studied. There aren't many laboratories actually looking at BMAA. They're probably, in addition to UNH, there are several dozen laboratories across the country, but not many. And there's the potential linkage with ALS and Alzheimer's that makes it a special interest. And then anatoxin, we just started working with anatoxins. They've been known for some time, and they're called the very fast death factor. And for good reason, that within hours of being consumed, they often cause the death of cattle or dogs very often. Hundreds of dogs have died from the anatoxins, and other wildlife, of course. So that's one we're quite interested in and have just begun looking at, both in the water as well as in the air. So, you know, where are the bacteria in a lake? If you had to go to a lake and find cyanobacteria, where would you look? Well, it turns out you could look practically anywhere and probably find them. They're in the open water. They're in the bottom sediments. They're attached to plants, attached to rocks. So some of these are highly specialized. The ones sometimes that are attached to rocks you don't find in the open plankton. But most of the work that's been done with cyanobacteria in lakes has been done with the open water. Just keeping in mind that if you're interested in cyanobacteria, there's plenty to do because all these other habitats need to be studied in more detail than they have. Okay, so we're going to focus today on the open water so that we'll talk about that. One of the first things that's really interesting is that you can't tell from a lake, looking at a lake, whether it has a problem with cyanobacteria. And this is a lake, Willen Pond, which is right in the city of Dover. And we took a series of samples in that lake. It's about 8 meters deep where we were, maybe 9. And each one of these jars is taken at a different depth. And if you notice, it doesn't take a great deal of skill to decide which one has the cyanobacteria. And that is the 7 meter, there was a layer of a particular cyanobacteria, which is Oscillatory, has a very low light requirement, so it can live with low light. And in a clear lake, there's plenty of light at 7 meters. So it was specializing in that depth. Now it can move up at times. So we may see it at the surface, say in the fall. So, but if you looked at the lake, you would know that there is a potentially a toxic layer in this deep water. What we've discovered, more recently, is the importance not only of those which come up and cause surface blooms, but of this other group that John had mentioned, which is the Picoplankton, pico meaning tiny. And these are less than 2 microns, which sort of gets down to the limit of resolution of most of our light microscopes. They can see it, but you can't see great enough detail to tell much about it. So they've been overlooked largely. The picocyanobacteria have been referred to as the most numerous, most abundant photosynthetic organisms in the world. And one of the reasons is they dominate the oceans. So that's where they were originally discovered back in the late 1980s, which is interesting, not all that long ago. And they were discovered using microscopic techniques that we now employ, which is using fluorescence. The other group, of course, is the bloom-forming cyanobacteria. We affectionately call them the BFCs. And they're the ones that we see as sort of surface scum on lakes. They can become highly toxic, in part because they live a long time, so they can store toxins. The little tiny picoplankton produce toxins, but their turnover rate is such that they only live probably a few days to a few weeks at most. So there's not much opportunity for much storage, but they turn over at a high rate. So they're important for the whole ecosystem. What we've also sort of developed then is a model of how this works in the lake. And so both of these are important in the lake. And we won't really understand the cyanobacteria problem until we look at both of those. So if we take a look at the picocyanobacteria, which sometimes we refer to as the edibles, simply because they're easily consumed by the grazing zooplankton, such as Daphnia and copepods. And these then are passed up the food chain into the fish and on into whatever happens to be the next highest trophic level. In this case, it's showing loons, but it could be just as easily bigger fish, or it could be otter, or what mink, for example. So all these then are part of what we consider to be sort of a classic food chain. So the importance of the picocyanobacteria is that they feed into the food chain well because they're edible. The big BFCs, bloom-forming cyanobacteria, are generally too large to be eaten. So they can develop a long time because they're not being grazed upon heavily. So they end up then having excess numbers that come up at times and float at the surface. Many of these have gas vesicles that collect gas then that brings them to the surface and where they stay. So this is simply a small video to show a grazer such as Daphnia here. And this is feeding on some cyanobacteria and picoplankton in here. What you're going to see is it kicks out with that claw. It can kick out these large lumps. Those actually are Vornicinia, a large colonial cyanobacteria, so that rather than eat it, they reject it. Also, these filaments, which is Dolichospermum, it produces lots of anotoxin at times. There it shows it working on Dolichospermum, but it's going to kick it right back out because it interferes with this apparatus. So this is why the tiny picoplankton then have a different role than the large bloom-forming plankton. So if we look at the effects of the cyanobacteria, not only on humans, but let's look at wildlife first. The model that we would use is basically the tiny picocyanobacteria go into the zooplankton, and then these are transferred up into the consumers of the zooplankton, the fish, which go to the, in this case, the loons, in part because there are many loons in our lakes, but also we're quite interested in them as a species. And we did a project in which we collected loon blood from a large number of lakes, 10 different lakes. And these were captures of loons then at night, where the blood then was taken from near the foot. And essentially, we developed a technique to analyze the blood for BMAA. Actually, I don't think anyone else has used that yet. So we were able to then ask the question, if this model is correct, that is that when BMAA is produced by the picocyanobacteria, it's transferred. We said that the greater that transfer, the larger the levels of BMAA in the blood of the loons should be. And so this is essentially a model, a mathematical model that shows the ratio of the zooplankton BMAA to the phytoplankton BMAA. And that means if it's large, that more is being transferred. So when more was being transferred, we got higher levels of BMAA in the blood of the loons. So that was the first step in just saying, does it really work this way? And that seems to at least support that model. We did a study in 2013 on Providence Lake, which is part of Awa, I believe. And it was a food web study. And we went out and collected samples of plankton, and we also were able to get some fish samples. And we looked at both the liver toxins, microcystins, and BMAA in the food web of Providence Lake to see, in fact, if this accumulation is occurring as we were suggesting it would take place. When we did that, these were the results. It showed that the microcystins were present in the phytoplankton, which is the cyanobacteria here. That's the green bar. And then this is the zooplankton, was higher. So there was some accumulation, but surprisingly, the largemouth bass didn't follow and didn't have higher levels. In other words, it was not accumulated, and that was not biomagnifying. However, when we looked at the pickerel, for some reason, seemed to show the greatest biomagnification of these microcystins. So if you were eating the pickerel, you would have gotten high levels of microcystins from eating it, whereas in the bass, the levels would have been lower, at least. They were still elevated over what was in the phytoplankton itself. The mussels in the lake, we didn't have much of a sample, but the ones that we did, even in the foot of the mussel, we got fairly high levels of these toxins. So again, a good reason not to be eating freshwater mussels. Now, we did another study in 2020. This study was actually precipitated by a loon called Luna. I don't know, has anyone here heard of Luna? It was the baby loon that occurred in Providence Lake. For years, people had been waiting for the loons that nested there to have a baby, and finally it did. And after it got to the point of being able to fly, but it was still on the lake, it suddenly died. So we were interested in seeing, could we see any evidence that maybe the toxins were involved? We really didn't know. And so I was teaching a course in ecotoxicology at the time, and a student from Holland, her name was Michelle Verstraeten, actually was in the class at that time and took an interest in this as well. And so what you see in the right-hand corner is actually Michelle doing a necropsy as part of our course on some aquatic birds. And so basically these are her data, and she wrote a paper on this, which is available if anyone would like to see it. It's called The Transfer of Neurotoxic BMAA from Cyanobacterial Blooms in a New Hampshire Freshwater Lake Food Chain. So she basically, as part of the course, wrote this up, and we put it into publication. Sorry. One of the things we found there was that the BMAA in the phytoplankton was quite low, but basically all the other forms were biomagnifying, which is quite interesting. So when we looked at the zooplankton, it was at least 5 times as much as the phytoplankton, and then the essentially common loon, which is CL here, the lungs and the liver were quite high. And surprisingly, the lungs were high. That raised some questions about whether the loon had been accumulating some of its toxins through breathing them on the lake. So we can talk about any of this later in the discussion section, but I certainly, there are a lot of interesting questions raised by that. We now think that the loon itself may have died as a combination of factors. The toxins could have been part of that, but also it could have been wounds inflicted in part by other loons. So let's talk about human health. Well, I thought before we describe how terrible the cyanobacteria are for human health, and that's pretty easy to do, I thought I would just touch upon something that we don't talk about much, which is possible health benefits. And I found a paper that listed 34 different species of cyanobacteria that are being considered as possible drugs that for cures for diseases, for tumors, for cancer, for various things. So because they produce such unique unique molecules, chemicals, it has attracted the drug companies to look at these as possibly offering cures for some of the diseases that we have. So who knows, some of these may actually then be helping us. So back to the human health effects. Well, one of the things that we often can see very easily in a system, this is a picture I took at the Old Durham Reservoir last year, and it shows a cyanobacteria called Oscillatory. It has this sort of wispy look to it, little threads almost, and it breaks up very easily. But if a dog were to get into the water, or a child, and actually consume just a portion of one of these colonies, they could get a lethal dose of anatoxins, the very fast-acting death factor. So this is one of the leading causes of many pets and essentially livestock drinking, where these are at the surface and they're easily consumed. So simply looking at a system, you can tell the health threat is there and obviously should be avoided. Sometimes it doesn't always work that way. And knowing when to avoid things, you might think that makes a lot of sense. But let me just show you, this is a lake in Massachusetts that we were studying. And this was mainly Microcystus on the surface, I believe. So it just gives you an idea that many times people don't take precautions when you might think they should. In this particular case, we found levels quite high in both BMAA and microcystins in this particular lake. So they were, the BMAA was the highest we'd seen in any of the lakes around. It was nearly 25 micrograms per liter. So that's quite elevated. So what other evidence is there that the cyanobacteria actually cause problems? Well, one of the things I wanted to just mention to you is that what got me into the looking for BMAA and its possible relationship to Alzheimer's and ALS was a phone call from a doctor at the Dartmouth Hitchcock Medical Center who was interested in Mascoma Lake. And he said that they had found a cluster of patients there. And so that led to us investigating it and writing a paper about this. The paper was by Tracy Collar et al., which includes me, as well as Amanda McQuaid, in fact. So we both, Amanda and I, were looking at that lake because we were called to see, could there be cyanobacteria in there that could be accounting for clusters? Then there's a couple of studies that are really quite interesting, and maybe you've seen these before, I've talked about them before, but I just wanted to, this is pretty strong evidence of something going on here. And that is a study by Zhang in 2015, which was an epidemiological study using essentially a satellite imagery to judge the quality of lakes and reservoirs, and then look at the distribution of non-alcoholic liver disease. And he found a very convincingly strong correlation between the water quality and the blooms in reservoirs and the high incidence of liver disease. Presumably, then that would be a result of microcystins in the water, but it could be some other toxins as well from the cyanobacteria. And then a more recent study, 2016 and 2017, there are two publications on this. And this was done by Torbeck et al. And I did participate in this study a bit. And it looked at New England and looked at clusters of ALS in New England. And with a little more refined use of the satellite imagery, they were able to estimate the amount of cyanobacteria in these lakes. And they found a strong correlation then between the water quality and the cyanobacteria in the lake and the likelihood of belonging to a cluster of ALS in a particular region. One of the interesting things, however, is that their study suggested that the sphere of influence of the lake extended out at least 5 to 10 kilometers or more, in some cases up to about 30 kilometers. So in fact, it wasn't just if you were recreating on the lake, but if you lived in the vicinity of a lake, which then raised questions. Well, why would that be? How would you, why would a lake be a threat to you if you didn't go visit the lake? And then that brought up the question of possibly the toxins were somehow airborne. So in 2013, Elijah Stummel, the person who called Amanda McQuaid and me and initiated our interest in this problem, he published a paper entitled Aerosolization of Cyanobacteria as a Risk Factor for Amyotrophic Lateral Sclerosis. And that paper then got us really seriously thinking about the need to be monitoring not just the water quality, but the air quality. He also, at that same time, began a study looking at some of the patients at Dartmouth-Hitchcock who had died of ALS and compared the presence of cyanobacteria in their respiratory tract with those of comparable age but had died of some other illness. And this was authored, this paper, by Fasaponte, who was a student at Dartmouth at the time. And it's a very interesting paper. I suggest you may want to take a look at it. But one of the things it did is they sent some samples to our lab to see if we could identify cyanobacteria in the tissues that had been taken from these respiratory tracts. And sure enough, it did appear that there were cyanobacteria there and not in the tracts of people who died of other diseases. So that's just another interesting tidbit. But it did raise the question then of whether we needed to be monitoring the air coming off of these lakes. So we began then developing, Amanda and I developed a method, and we further improved that method. And this is Kate Langley, who did her master's with me about 2 years ago, collecting aerosols at Lake Mascoma, which is in Enfield, one of the hot spots shown on this map. Each one of these dark spots here is made up of dots. Each dot represents then an excess case of ALS. So in the case of Mascoma, it was approximately 20 to 25 times what you would expect if the cases were evenly distributed. So that's how that was done. And you can see it for Vermont, New Hampshire, and Maine here. So there are other hot spots. And so that's one of the interesting questions then is this being transmitted through the air? And so what you see here with Kate Langley's aerosol collector is that essentially we're sucking the air just above the water surface, putting it through filters, and now we also put it through a water trap, excuse me, and collect then the cells and the toxins themselves that are being emitted by the lake. So now we look at a lake very differently, because if you look at the lake, obviously you don't see this happening. But if you put these collectors out there, it happens. So I'm trying to visualize what this might look like. And so what I see is, if we could see it, this is what our lakes would look like. And these white dots coming out of the lake would represent then the cells, which we know come out in the thousands. And then the sort of the smoky stuff would be the toxins themselves, which also come out of the lake. And so this is a view then of what the lake would look like if you could see these toxins. Okay, so let's get back to the health risk just a bit more. And so if you look at the risk then of going in the lake, and when cyanobacteria are in high numbers, where does that risk come from? Well, dermal penetration is one. Obviously, the surface of the skin is exposed to the water and the toxins and cells that are in there. But fortunately, most of our skin has a fairly impervious waxy layer. And some of the cyanotoxins can cause dermatitis. And so they actually react directly with the skin. But most of them don't penetrate the skin as rapidly as they do through the other two methods. So I put this as a dotted line because actual accumulation of toxins through the skin is probably not as important as the other two. So, and that is through oral or ingestion. And ingestion is interesting because the EPA spent a lot of time and money estimating how much water do people ingest whenever they go swimming. And the averages for this are that children, on the average, drink more water when they're swimming per swim than adults. And so, in fact, it's basically twice as much water, 47 milliliters, or about 1.6 ounces. And for adults, it's about half of that, 24 milliliters per swim. So we may not think we're drinking water, but we are. Well, is that important? Well, one of the sort of limits that we've set for drinking water is somewhere about a microgram per liter of microcystins. And the microcystins then at a microgram per liter that's pretty much for adults. For children, the older level was 0.3 micrograms per liter. So if you, however, if you were to be swimming and encounter a clump of cyanobacteria on the surface, it's a very different condition, because the water itself may have far less than a microgram per liter in it, but a clump of it, we've measured clumps with as much as 100 micrograms per liter, 1,000 micrograms per liter, even more than that, in clumps of this cyanobacteria. In which case, then you could get a toxic dose of that by just drinking the amount that you would get during the swim. So I think the danger there comes not in ingesting the water per se, but being careful that there are not clumps of cyanobacteria floating around when you're swimming. The third way that the cyanotoxins get into the body, of course, is through inhalation. There's far less known about inhalation, in part because very few people measure the aerosols that are coming off of lakes. That's something that we are very excited about at UNH, because we are learning more and more. We just had, in fact, two very exciting teams of undergraduates presented posters at the UNH poster session on Saturday. And they were both looking at aerosols and factors that regulate the aerosols coming off of lakes. Very exciting work. And I think it's groundbreaking what these undergrads were able to do in our lab in just a short time. So we're very excited about that. The interesting thing about inhalation is we think about drinking water and we talk about, oh yes, you drink maybe 1 or 2 liters a day, some people more. But with air, of course, we consume much more air. And a meter cubed is actually 1,000 liters. And so on the average, adults, depending on the activity level and the size of the adult, it's basically somewhere between 10 and 30 cubic meters of air we consume every day. So that even small amounts of the toxin in the air, both are taken into the body, but also into the lungs where transfer of toxins can occur relatively easily. So that would be the equivalent of somewhere between 10,000 and 30,000 liters consumed, if you put it in those terms. So this is then what you have to be concerned about if you go into a lake where there are excess cyanobacteria. So the 2 basic types of cyanobacteria that we have are the big things, which are the BFCs. These are the ones that come to the surface, and the very small ones. Oops. So basically, I wanted to show that we already did this. Hold on just a second. So what I wanted to do as the last part of this is, what do you do about if you have cyanobacteria, you suspect you may have cyanobacteria in your lake, or you simply want to know, do I have cyanobacteria that I should be concerned about? If you said, do I have cyanobacteria in the lake, or someone asked you, you should be able to answer them pretty easily and say, of course you have cyanobacteria in your lake. Everyone has cyanobacteria in their lakes because they are part of the lake system. The question is, are they a problem? So in order to answer that question, you have to start with what I consider to be a 3-step program, which is just getting to know your lake and getting to know your lake community. So basically, step 1 is to do an assessment of the cyanobacteria and the lake food web they exist in. So the question is, what types of cyanobacteria are in my lake, would be the question you're asking. And is the food web in the lake in a healthy balance? In other words, is it operating the way it should be to keep things from building up in the lake? So how do you do that? Well, one of the things we've taken a great deal of time and effort trying to develop is a technique that separates out cyanobacteria from other plankton and separates out the zooplankton from the phytoplankton. The problem is that they overlap in size, so you can't put them through a mesh and separate out the cyanobacteria or the phytoplankton from the other forms. So Amanda actually was involved in this, and I don't think that is Amanda there, and I think it's one of our helpers there out on Christine Lake, probably, because it looks so cold. That's up in Stark, New Hampshire. But basically, what we've created here is a separation bottle. This was the earlier form of what we did. And basically, it's dark at the top and light at the bottom. And the idea behind this is quite simple. We know that cyanobacteria blooms occur generally when you have calm water, very little wind, and you have basically cloudy weather, so low light, and that they move toward the surface. The gas vesicles pull them toward the surface, and they form this scum. So that's why you can find the cyanobacteria on a lake in the morning, say, after a night that's been very calm. And then as soon as the wind picks up, they're gone because they get mixed back in. So all we did is create the conditions for blooms to occur. The lighted bottom, however, has another purpose in that the zooplankton, such as the crustaceans here shown, the Daphnia, they actually are attracted to the light. So they go down, and then the cyanobacteria go up. And so if you leave these sit quietly for about a half hour or so, depending on the size bottles that you're using, then you get this separation. So you can go out to a lake that has no bloom, and if you collect a sample of plankton from that lake, put it into this device, it basically will allow you to see, do I have BFCs? Do I have bloom-forming cyanobacteria that, given the right conditions, would form a bloom in my lake? So you're artificially creating a bloom, essentially. So this is the device that Amanda McQuaid used for her thesis work. And then we got, you know, idle hands in the laboratory always create new problems. And so what we did is we tried to figure out a better way to do this, maybe a smaller one, because this actually in a canoe took up quite a bit of space. So we developed what we call the Zapper, which is really the same thing, only a little more sophisticated, perhaps. We called it the Zapper because it's the zooplankton, phytoplankton photoresponse. That's what the Zapper stands for, plankton separator. And so this is showing some students in our classes that are operating these, and you can see the light at the bottom. This is sometimes done with a flashlight, or you can simply leave it out in the light, and the zooplankton then fill up the bottom, and in this dark container at the top, the phytoplankton go, and then you turn a knob here and essentially separates the two. Then you have the two fractions. So why would you want to do this? Well, basically, using the Zapper then would allow you to concentrate the bloom-forming cyanobacteria so that you could examine them and identify them. And so if you went out every, say, 1 or 2 weeks and collected samples like this over a summer, you would actually have a very, very good picture of the types of cyanobacteria in your lake. And knowing something about those cyanobacteria, you could also say, what are the likely toxins that these produce, if they are toxin producing? So then you would be able to say even more than just, I've got cyanobacteria, who are they and what kind of toxins would I expect? The other thing you can do with that is now we save this zooplankton fraction, and that concentrated fraction then, we measure the size of the crustaceans in there. And they are actually a good measure of the ratio of panfish to predator fish or game fish. So you can say something about the balance of your food web by looking at the size of these plankton. If you have overfeeding on the zooplankton because there are too few large fish species present, so the small ones that eat the zooplankton are too abundant, then the food is not being channeled and the energy up into the higher trophic levels, and you'll be able to see whether that is an issue in your lake. And then that can also cause problems with cyanobacteria, having that imbalance. So you would have to learn to use a microscope, which we offer as part of this program, and then you would buy the Zapper. And with the Zapper, then you would identify the phytoplankton online. There are several ways you can do this, but we have a simple online key to what we call the dirty dozen, which is the cyanobacteria that are commonly found in lakes that create problems and are often toxic. So that's an easy to use key. So I just wanted to show you how simple this Zapper is, just so that you get some idea what it looks like in use. And this is an EPA video they just recently made of what we now call the pocket Zapper. We weren't satisfied we'd created this previous Zapper. This is now one that is even smaller, so we call it the pocket Zapper. Sounds like something you'd see on late night TV.
We'll demonstrate how to take a sample of cyanobacteria from the Zapper. To do this, you will need the Zapper, your collected sample materials, And gloves. Remember to always use gloves when handling cyanobacteria. Prior to storage, make sure you rinse the zapper out with tap water. Doing this between sites will help prevent cross-contamination. Take your collected sample material and pour it into the zapper until the zapper is almost full. Place the top back onto the zapper and store it in an upright position for approximately 30 minutes. This will allow the bloom material to collect at the top of the zapper. Then, use a pipette to take a small sample of the bloom material so you can analyze it using the microscope or the fluorometer. In this video, we'll demonstrate how to take a sample of...
Okay, so that gives you some idea of how simple this is to use. And basically, when you take that sample of the BFCs which you have there, you can put it under the microscope and identify what species you have. And then also with that same sample, you can put it under a pigment analyzer, which will tell you how much biomass is there. So that's going to be, I'll talk about that in a couple minutes as an important part of what you can do as well. So then the other thing we are proposing that you should do is look at that food web, because that may well tell you have a problem. Many times, if you have too much fishing in the lake, for example, fishing derbies, you can remove a lot of the predator fish. And what does that do to your grazers that keep the lake cleared? So here's the just a little movie we made actually about showing a time lapse of how quickly you can see these white things coming down. This is the zooplankton streaming down into the bottom part of the zapper and collecting down there. And these are then largely copepods and daphnia. And then those we would put under the microscope, and you can measure the body lengths. And we have an equation that takes body length and converts it to the fish in the lake. That is to say, what is the balance of fish, the panfish to game fish in the lake? So that's kind of a fun thing. Most of all, you just get to know your lake much better, and you get to learn about things that live in your lake that you may never have considered. So step 2. So that could be, if you wanted to start this program, and maybe even the first season, you just wanted to start it off by learning what's there, that's a perfectly good way to do it. And the second step then is to look at the toxicity, because ultimately, one of the things that most people are concerned about with cyanobacteria is the fact that they're toxic, or they can be toxic. So one of the things we started doing with our program down in Cape Cod area is we began correlating the toxicity with cyanobacteria pigments. And so we now are using the pigments as a way of identifying cyanobacteria. And so that's a much simpler way in some ways. So the pigments we're using are called the red pigments, phycoerythrin, shown here, and the blue pigments, phycocyanin. These are more or less unique to cyanobacteria, and they help collect light for cyanobacteria that algae in general don't have. So they can only collect it with the light for the chlorophyll. So we're using then, especially phycocyanin and phycoerythrin, these what are called accessory or antennal pigments, as a way of assessing how much cyanobacteria is present. It's much better than chlorophyll, because with chlorophyll you can't differentiate between cyanobacteria and any other chlorophyll-containing organism. So these are the 3 pigments, and they're very simple, because with laser beams using the appropriate colored light, you can then detect the phycoerythrin, phycocyanin, and chlorophyll as well, almost instantaneously. So we're trying to develop then thumbprints essentially for different lakes and different types of cyanobacteria, so that using this, we can differentiate between the picos, the BFCs, and other phytoplankton. So how do you measure it? Well, it's measured very simply with a fluorometer, what's called a handheld fluorometer. There are a couple of different varieties of these. This is one that we use a lot in our lab, but not the only one. And basically, with this, you can measure then the amount of these three pigments, or at least the phycocyan and phycoerythrin, very quickly. And why do you use this? Because it's quick, it's really simple to use, and it's very inexpensive, whereas it would cost you easily several $100s to do the toxicity tests that are needed for samples that you have. Here you could basically, once you've invested in the fluorometer, you basically can do the pigment measurements for very little at all. So the last step that I want to mention, and I'm going to try to end very quickly here, is step 3. And that is, now if we have got these measures of the cyanobacteria pigments, we identified who is there. Now we can actually use that information for what we call cyanocasting. That is, we can forecast two things. One is we can forecast the toxicity of a particular sample. And then also we can begin looking at growth rates in the lake. So let's look at this. This is some work we did down in Cape Cod, and a lot of this was done by Nancy Leyland, who's played a big role in developing this concept. But basically what we found is that if you looked at the PC, which is phycocyanin, that's the blue pigment, and MC, which is microcystins, that's the liver toxin, what you find is that there's a nice relationship shown here in the red values. Whenever you have a particular cyanobacteria called Microcystus, very common in many of our lakes, when it's the dominant, this is what you find, this relationship. Whenever you have Dolichospermum, which used to be called Anabina, when it's present, it doesn't produce as much microcystin. So you need a different relationship for it. And if it's mixed assemblages, it's somewhere in between. So we said, if this is really quite interesting, because this would mean that at least we could get an estimate, a first order estimate of toxicity by just taking a measure of the pigments present, and we should be able to predict how much of the toxin is there. We tried it on different lakes in the Cape Cod region, and as well as other New England sites, Silver Lake of Hollis, New Hampshire, and Lake Massabesic in which is the big drinking water area there, and Long Pond in Centerville. And so when we looked at these others, they fit the same regression essentially. So this gave us encouragement that this could be used. This is still, New England, and we don't know whether there are regional differences. So what we'd like to do is, if you were to do this for your lake or your lakes, say if Agua were to take this on, you'd want to see, do your lakes operate in the same way? When you have microcystis, do they give you very high levels of the toxin microcystin? And if you have a Dolichospermum in your lake as a dominant, does it actually then fall into this lower level so that it produces less of the microcystins, but in fact, it's also producing another toxin, which is the anotoxin. So we want to do this for at least the microcystins, but maybe for at least microcystins and anotoxin. So developing this for both of these gives you a very powerful tool that you can then go out to your lake if you have cyanobacteria blooming, measure the levels of the pigments, and from that say, well, according to this, we're above the level that is safe for drinking water, let's say, or for recreation. And so we should have this water tested with another method. So it's a way of giving you a tool that you can then begin to estimate on your own what the toxin levels are, and then you would want to confirm them with another method. The other thing you can do is that, this is interesting, is that with measuring the pigment levels, every, say, week, you can look at the rate of change in the amount of pigments that are present in the water. And that's a measure of the growth rate of the cyanobacteria. And what we found here, which was quite interesting, just to show you one use of this, is that it shows that the microcystin production, how fast it was producing the microcystin, was positively correlated with its growth rate. That's very interesting. That matches actually what people have found in the laboratory. This was all measured with field samples. But what it says is that when cyanobacteria are happy, they produce toxins. They don't do that as a swan song whenever they're dying. They actually produce it when they are most energetic. The other thing it does is getting the growth rates in your lake can allow you to predict when the growth rates are a predictor of blooms to come. So in other words, when the growth rate hits a certain number, we found that within a certain number of days, we can expect to see a bloom given calm weather. Now, this is especially helpful for people who are in the drinking water business, because they really need to know when a bloom is going to occur, because they may have to change their water source or move their water source from maybe the surface waters to the deeper water. So, but also for recreation, it can be very useful to know that. So that's another use of this information. So if you need follow-up information, you can contact me, obviously. But I also wanted to just introduce Amanda McQuaid. Amanda, are you here? If you can unmute yourself.
Yes, hello.
Hi, so Amanda worked for DES for several years. How many years did you work there?
About 5.
About 5 years, and she actually talked to this group, I think the Water Talk last year. But in the meantime, she's actually been hired back at the University of New Hampshire as a professor in the biological sciences as well as cooperative extension. And she is part of our ecotoxicology lab, but also become the director of the Lakes Lay Monitoring Program. So if anyone here were interested in maybe learning more about some of these ideas of what you could do and maybe begin developing a cyanobacteria monitoring system that gives you more powers than we've ever seen before with some of the things we're doing, either I or Amanda would be happy to help you develop this. So I'm not selling it, but if you are interested, we'll give you more information. And where you can get the supplies needed, and maybe even, we've talked about maybe offering a workshop on this sort of thing. So this is all kind of new stuff. So with that, I went over a few minutes, but for me, that's not bad. I told John I would finish at 7. So 7:08, I feel pretty good about that. How about you, John?
Oh, that's fine, Jim. I think everyone wants to hear you talk, so that's no issue there. Okay. There are a ton of questions, so if we can keep you around for a few more minutes, we'll definitely we'll get to as many as we can. Just so let me know if you do have to go. But I do want to touch on a couple of questions. I'm going to kind of work backwards from the most recent questions back to the beginning, since it's you sort of culminated in what's really important here, and talking about how people can get involved and understand how to do it on their lakes. So one of the first questions we got when you were talking about pigment levels, is that specifically, I think you did mention this, but just kind of a clarifying question, do the pigment levels refer to how many cells are in a bloom, and then how does that pertain to at what level would that constitute like a swimming advisory or something?
Yeah, like well, that's really a very important question. One of the biggest difficulties we have in trying to determine how much cyanobacteria are in a system is what measure do we use? And because some of the cells could be Maybe some of them are 10, 12 microns in diameter, and others may be 1 micron in diameter. If you look at that as volume, and the volume of a cell is the cube of its radius, then what we see is that all cells are not created equal. So if we just count the number of cells, it gives us some idea of what's there, but it's not a very quantitative estimate because it depends on what kind of cells they are. And so with the pigments, this is yet another opportunity to, because the pigments, we're using that as a surrogate for biomass. So we think it's more related to the size of the cell than the number of cells present. So basically we're substituting the pigment as a measure of biomass. People have calculated volume, but of course, even when you get the volume of a cell, you don't know whether the cell is full of something or whether it's not, because cells, when they're very healthy, tend to be full of protoplasm, but when they're not, so just looking at the cell and counting it doesn't get you much more. So there's no perfect way of doing this. And the advantage to what we're doing with the pigments is the pigments, you can get relatively accurate measures of how much pigment is present. So, and it's very quick. You can do that within minutes. And as anyone who's tried to count cyanobacteria, and I know Amanda has done a lot of this at DES, it's very demanding, and time consuming, and sometimes frustrating because it depends on the types. If you get a big colony of, say, Microcystus, for example, and there's cells all through this mass, it's hard to focus on everything. So you have to estimate. So it's not, there's no one way is perfect, but we think this is probably as good as looking at some of the others. So that's the long answer to your short question.
We had another question about private drinking wells. So is there, or what is the risk of intrusion into private drinking wells that are, say, like within 100 feet of the lake, and can, like, specifically BFCs get in, or maybe the Cyanobacteria? Is there any research on groundwater access in proximity to surface blooms?
Yeah, there's been a lot of interest in that. And some work was done in the southern states where there was largely sandy soils, and it was found that they could detect the microcystins as much as maybe a quarter of a mile from the source in fairly large quantities, actually, so that these are pervious groundwater systems. The problem with New England and New Hampshire, especially in Maine, is that, as you probably know, if you've looked for drinking water in this, a lot of it runs through little veins, and you either hit it or you miss it. So the same thing could be said of water intrusion. So I think the only way to be happy with that, and we've tried doing that on a couple of lakes, and sometimes we found something and sometimes we didn't. But I think the easiest thing would be to get water from your system and see if you can detect cyanobacteria with the pigments. So that would be a really simple way, and because it is possible that the pigments are getting through along with the toxins. So what we're finding is we're getting pigments now in our aerosols as well. These small molecules may be good trackers of where the cyanotoxins are going as well. So if you find the pigments, you definitely could say, well, we've got a problem. There's definitely, if you're close to the lake and you're finding cyanobacteria pigments in your water, and it's a water that's not exposed to the light, then it's coming through the groundwater most likely. If you don't find them, you may have to test for the toxins themselves.
So you would recommend folks testing well water near the lake for pigments?
I would start with the pigments. It's the lowest hanging fruit.
Yeah. Awesome. So on the aerosol subject, that's a pretty big revelation there. It's really interesting that the research you guys are doing. One question we had was, how far from the lake could aerosols be a concern? I noticed the picture of a sort of a radius around Lake Sebago up there.
Yeah.
And does that change based on how forested the surrounding area is, or, you know, whether it's cleared or open or developed versus forested, or is there just a general radius around the lake where aerosols can be a concern? And the other half of that is, how exactly do aerosols, how do Picocyanobacteria get from the water into the air, and how are they traveling around?
So I would say I'd like to find out who asked that question. And then I would invite them to come to UNH and do a PhD on this. Because obviously we don't have the answer to that, because that's one of the, there was a student at Dartmouth who contacted me last month, and she was doing a project as part of a CDC grant, the Center for Disease Control. And what they wanted to do was estimate the distribution of toxins in the air coming from a lake, and exactly the questions you're asking. And so the problem is, it would all be very easy if it weren't for wind, you know, because it would be just a diffusion gradient. So that would be something that would be relatively easy to model. The hardest thing to model is wind action. So what we're doing with the work on Cape Cod, I've got a student, Haley Carter, who's just finishing up this spring, and she's been studying 3 different toxins in the aerosols and finding all 3 toxins, the microcystins, the BMAA, as well as the anotoxins in the air. And she's going to be estimating sort of a loading, an atmospheric loading from this lake. How much does this lake contribute to the atmosphere? Because we can do that. The next step, which is how do we measure the risk if you live, you know, near the lake versus maybe a kilometer away from the lake, that's a harder thing. And that's going to take some careful study. And I don't think it's something we should shy away from, but that's where we're at. That's why I'm sort of half jokingly saying, you know, if someone's interested in that, we need people to study it. And so we don't know at the moment. One of the things I thought I would like to try is I'd like to just go around with a vacuum cleaner and vacuum people's houses. And the reason for that is that one can assume that if it's dry, some of these toxins are stable, and they'll stay in dust particles. So we might be able to track the amount of exposure by looking at the dust in people's houses at different distances from the lake and different positions around the lake. So it's just one of many, many ways you could look at this. The other, which is also possible, and they started this in Florida recently, is to look at blood samples from people who live around lakes, and just see if the blood, like with the Loon work that we did, which was very promising, could we measure then the level of toxins of these cyanobacteria toxins in your blood, and try to correlate that with location around from the lake or around the lake, because there could be upwind sides of the lake where you're protected. So it's not just distance, but on the scale that this study was done by Nate Torbick in New England, it looked like it was something about 10 to 30 kilometers was where they could see an influence of the lake. Mm. Yeah.
But probably dissipating over that period.
Yeah, and one would assume that closer is probably in general going to give you higher exposures.
Well, I wasn't going to disclose, but that was actually my question. So if you'd like to,
there you go.
If you can help me bypass the admissions process, I'd be happy to discuss my future PhD in another call.
Not a problem, John.
All right, moving on from that lovely topic. let's see here. Talked about that. Earlier on, you mentioned a study by Zhang, I think was what it was. And somebody asked if in that study, how specifically cyanobacteria was getting into people's bodies. So I know you mentioned a couple of ways that it does happen, but were you aware of what method or what they were studying as far as how people were exposed to cyanobacteria?
Well, the Zhang study was strictly epidemiological. In other words, they simply looked at the incidence that they from the records of people who had died from liver disease not related to alcoholism. Okay, so it's called non-alcoholic liver disease. And then they correlated that from the distances of those cases to the quality of the water based upon satellite imagery. And they were able to discern that lakes that had some cyanobacteria from those that had full-scale blooms. So they felt that the blooms were the worst condition. And that's very interesting in itself. You know, the surface blooms could be either providing more contact. We're not sure that they actually create more aerosols, but that's just one of the many questions we have. And one of the, you know, there's always I would say that there's always motivation that needs to be disclosed. And my motivation in part for wanting you guys to get involved is we want to learn more about this. And we learn by doing. We don't learn by talking about it. You know, if you get out and you contribute to a database that actually it may be of more value to science than it is you. We don't know yet. But just by testing some of these hypotheses, and this is, you know, these are all ideas that are just developing. And so I think you're part of a research team when you participate. And that's sort of the motivation that I think Amanda and I have. We want people to, that's been the motivation for lakes monitoring ever since we began the lakes monitoring in 1978. That we wanted to learn more about lakes in New Hampshire, and it's really helped a lot. But now the new questions are arising, and we still need to learn much, much more.
Yeah, it seems like you guys have done a lot of, you know, studies on a lot of lakes in New Hampshire. Somebody did ask if there's like a public source where we can view the studies that have already been done on lakes in New Hampshire for your work or for any of the work that's been done in this field.
Well, there's a couple ways to do that, and Amanda could address some of this because with DES, they do have information from the VLAP program, not specifically so much on cyanobacteria, although they may be getting into that more. But our Lakes Lay monitoring program does have the reports that have been done in the past. And again, many of those don't address cyanobacteria, but our own Center for Freshwater Biology, and Amanda and I are in the process of publishing quite a number of studies that we've had done at UNH dealing with lakes and cyanobacteria. We have some of them are already published, but and putting those, updating our online system. So we'll get that to you as soon as we activate that. We're also going to create a new web page for the ecotoxicology lab, and it will have its own. It will also participate in that reporting system. That question is a really good question because I think this is the most underserved part of most programs. That is the public knowing, and to me that should be the first thing we look at. I mean, the reason that citizen science exists is because citizens are interested in the answers to these questions. They're not just that, but that's the primary. So they want to know, what did we find? What's the answer to my question? And so we're hoping that our new ecotoxicology webpage will have tools on it that citizens can use to find things and also to make calculations to actually participate in what we're doing and hopefully get some of the excitement of what we're doing as well. Amanda, did you want to say anything in that regard?
Yeah, just that the DES has something called the Lake Info Mapper. So you can kind of scroll into your lake and see if there's ever been a recorded advisory with the state at least. And as Jim mentioned, our CFB website does have some pretty detailed New Hampshire lake studies that focus on cyanobacteria. So it would be more about kind of browsing through what we already have. But as Jim mentioned, we have a lot of studies we haven't posted. And also the Lake Slate Monitoring Program can also be found at the Lake Info Mapper. But our website, like Jim just mentioned, is in development. We're changing some things, so it might not link up very well right now. But if you go to the UNH library database and just search for a lake or a topic, those will show up pretty well, even through a Google search, I guess. So the UNH library database is also really great. And thanks for sharing that link.
Yeah, I was going to say, I put the Lake Info Mapper link in the chat for anyone to take a look. I've used it many times. It's a great resource. And you said there's also something available on the UNH library website as well.
Yeah, you'll be able to see any publications or theses that, Jim has been an advisor to. So there's a lot of students, of course, over the years. So it's a great way to just look. You'll also find the Lake Slate monitoring reports there, which will also be on the Lake Info Mapper, but the link might be broken right now, where you could see the VLAP reports on the Lake Info Mapper, or it'll have a link that says cyanobacteria bloom history. And again, that's just if it was an advisory, not even just a record. So there's a lot more to it, of course. If there's been cyanobacteria, probably most of the lakes have, but that's just a way to track if there's been a state advisory.
Great. Okay, I have kind of three questions that I'm going to lump into one. Some folks were asking just about the nature of cyanobacteria in the lake, and somebody said if blooms are mostly growing during the rainy season in the heat, why are we also seeing them in the fall and sometimes even the winter? And another part of that question is anything that, are we just looking for surface scum, or some people are just seeing blue-green like particles submerged in the water? So is that also likely cyanobacteria just based on the color? Does it have to be in the bloom or in that large sort of blue-green swath that we saw in some of those videos? I know that's kind of a disjointed question, but I was just trying to lump a few things together. So what kind of things are we looking for when we're identifying cyano, and then why are we seeing it in every season?
Yeah, well, let's start with that question, because one of the things that I mentioned is, where do we find cyanobacteria in the lake? And I basically said you can find them in all habitats within a lake even. And so many of the cyanobacteria are evolved. If you look at the phosphorus level in the water, it tends to be lower than the phosphorus level in the sediments, because the sediments consist of organisms that have settled and died and decomposed. So it's a compost pile in a way. So many cyanobacteria have evolved to take advantage of that and go down and rest in the compost, which is the sediments, and then rise to the surface whenever it suits them. So they can appear at any time of the year. We get sometimes coming up under the ice. Some of it may be induced by gas bubbles that develop within the colonies and raise them to the surface. We've had fishermen contact us and said, there's this, we punched a hole through the ice and this green liquid came out all over the ice. We went up and sampled it, and sure enough, this was in Concord, in one of the ponds right in the city, and it turned out to be aphanosomnin, and it had we got a 21-liter container of aphan. I'd never seen so much come out, just so thick, and we tested it for toxins, and it was producing the saxitoxin. So they can be toxic. They can come off the bottom, and they're just a lot of different places, and that's why it's hard to know exactly where they are. And we study the water and more than we do the sediments. So that if we're looking at just the water, we can be very surprised when we see them coming up. But we've learned now that some things, again, like the leachospermum, typically overwinters on the sediments, and then it will come up right at this time of the year, early spring, and very often come up and form a layer at the surface, and it goes back down. We don't know exactly what they're doing that time, but then that also can happen again in October, November. And we've had those happen in water sources, for example, where they wonder, where did this stuff come from? And then if you look at the bottom, you'll see it down there, but it's not visible if you're just sampling the water. So that accounts for some of what you're saying. Mm-hmm. The other thing is, how do you know if it's cyanobacteria? If it's just sort of greenish looking, and there are some algae that can give that appearance, usually in very eutrophic systems. Most of our lakes in Maine, New Hampshire aren't like that unless they have extreme activity around them. So usually when we see a concentrate of pigment like that, it's very often cyanobacteria. There's no guarantee that it is. And of course, if you were part of this program and you actually had a microscope and you had a plankton net, then it would be quite easy to answer that question, right? There's motivation.
That's probably a good segue into the next string of questions here. So I think, you brought up a lot of fascinating points tonight. And I think, some things that can be a little unsettling. And I think folks are really going to want to know what they can do and how they can participate and sort of what precautions they should be taking. So what's the best way to start getting involved with that 3-step process and get cyanomonitoring happening on your lake if you're not already doing it? And also the other half of that is, what recommendations would you make for people, that just what people can do in their own life on the lake, in their homes and on their properties? I know that's something I kind of touch on too with my organization, but if there's anything from your perspective, what can folks be doing? What should they be doing, and how can they get involved?
Well, this is kind of the same question that Amanda and I have been wrestling with this spring. She just started actually at UNH again. And so we're thinking, well, how do we provide more assistance to lake groups? And what role should the Lake Slay Monitoring Program have in all this? And she's in the process of, in our new laboratory, developing a microscope room where she'll be able to do more with microscopy, because we think we've underestimated the value of identification of the organism. As you saw from those regression relationships we had, if we didn't have if we hadn't identified the dominant species, those regression relationships wouldn't even be significant. But once we know what species are dominant, then we have a powerful tool. And so that's why we decide that's probably the first step, if you're going to do this in steps, of just getting to know your phytoplankton, getting to know your cyanobacteria. Now, who's going to do that? That's the question that arises. And some people may be motivated to do it themselves. It really doesn't take too much. You can buy the plankton net and the Zapper. Basically, those are the two things that you need. And then a microscope that we recommend some that are fairly inexpensive. They may be around $100. You can get a little better one. And in fact, we promote that as well. It's like bird watching. You're better to get a good pair of binoculars if you're going to become a bird watcher. In the end, it pays. But if you can't afford that, some of the inexpensive scopes are okay. And then you work with us. And that's the question. How do you work with with our group, for example? I mean, you could work with other groups, but we're going to be around. And the idea is, what role should we play in it? And we're in the, and I think this is fair to say, Amanda, we're in the phase right now of trying to figure out what that role is. Should you be able to send us a sample? Or better yet, take some pictures through your microscope, because these microscopes have cameras, and allow us to then communicate, as most advanced people do anymore, using just the internet. And so you don't have to bring a sample necessarily to the lab, although that's a possibility. But and I've been doing this with, for example, the Rhode Island Health Board down there has gotten interested in cyanobacteria. And for the past year or two, we've been going over their findings, and now they're really capable of doing the identifications on their own. And we've done all that just through through internet conversations. And we look at a species and then talk about why it's this one and not that one. And before long, and what you'll find is, for cyanobacteria, you're not talking about hundreds of species that you have to identify. Usually there are a dozen or so, a dozen or two maybe, that you really need to know. And once you know that, you're actually pretty well qualified. So it's not as daunting as it may seem. So I think the question is, what is our role? And what would you like our role to be, is the other question. If, in the ideal world, what would it be? Would you like to be able to come to UNH or send a sample to Amanda and say, can you tell us what's in this sample? We've always felt it's better to teach people to fish than to sell them fish. So I don't know. So in that regard, obviously we'd prefer that people get involved as much as possible, because that's another educated person in the world. We need more of those now more than ever.
Yep. I'm definitely excited to see how that evolves, because I think it's great to get as many people involved as possible.
But think about that, and from your perspective as a leader of a group here, and how we can best help that group. Amanda, do you want to say anything on that?
Not too much. I think what you laid out in the 3 steps is something we're exploring, and we might not be fully ready to support everybody this summer. But we would like people to become more interested in the plankton net and the Zapper, and using that on the fluorometer. So definitely reach out to me if you'd like to discuss this further, or Jim. I'll also say that DES is getting a replacement for the cyano position.
Oh, yes.
They start this Friday, and they're very capable. So they're a former Haney lab student, in fact. So we're very excited about that. And I can just see future collaborations with the state and these water quality monitoring programs to help out. So I think it's going to be a really good shift.
It's awesome to hear. In talking about different species, you mentioned earlier that I think in one of your slides said Cylindrospermopsin, I think that's how you say it, was a tropical species, but is now in New Hampshire. So are species migrating north, and what are there implications to that, or what's
that was the original interpretation. Of course, so much of this is the recent interest in cyanobacteria makes it difficult to know, because what we identified 20, 30 years ago was a small fraction of what we're looking at now, just because of the number of eyes that are looking. So we don't know whether it has moved north, or whether we simply are looking for it now. So it's hard to judge. If at any rate, many of these species we thought were confined to the south, actually cyanobacteria thrive under warm conditions in general, but they also can tolerate cold conditions. They are cold hardy, and it turns out that at least some species of cyanobacteria are the dominants in the Antarctic as well as the Arctic. So, but we do know that, and this is probably related to their history in part, when the Earth was much, much warmer, when the atmosphere was primarily, it was anoxic, and it was a much hotter place to be at that time. And some of that warm tolerance may go back to genes that were evolved in the early times. And they can live and grow well above 30 degrees, 35 degrees, almost 40 degrees. Some of the species we have in our lakes here, whereas most of the other phytoplankton drop out. So, those growth curves are pretty convincing. But I don't think that the appearance of species here necessarily that weren't seen earlier necessarily means that they're moving north. I expect that cyanobacteria are going to be enhanced by the global warming. I mean, things are getting better for them, yes. If we can just get rid of the oxygen in the air.
Pesky oxygen. So it's bringing us down. This might just be an opinion question, but I didn't want to exclude it, and it's relevant to like the human health and the wildlife health question. Would you recommend catch and release for fishing, not only for the health of lakes, but also as far as ingestion of fish and wildlife from a human health perspective, but also from an ecosystem perspective, as it pertains to like keeping these nutrients within the system or anything like that?
Well, I think it's not so much the nutrients. I mean, we now know, one thing we know now that we didn't 30, 40 years ago is how important top-down effects are in aquatic ecosystems. And manipulating the fish populations can have a big influence on water quality and cyanobacteria as well. So, it was thought before that the fish and game operated totally independently of DES because they were unrelated. Turns out they're really very closely related. What goes on with the fishing affects the other. And we're beginning to appreciate it. But back to your question, I think many systems run into problems whenever they have stunting of fish or the lack of the top predators in the system. And that comes sometimes because we focus our attention in fishing on the large species. So, returning the large ones back into the system generally makes sense, because that way they're going to keep the small ones under control and not over exploit the zooplankton. That's the biggest thing. There's a system, I don't know if you know Highland Lake in Maine, but it has alewife coming into it, and the alewife decimate because they are planktivorous, they decimate the zooplankton, and then they have blooms of Cyanobacteria. Not too many lakes have actual blooms where it turns green with Cyanobacteria, and yet they do, and that's because there's nothing to graze on them. And we're studying a lake in Cape Cod that's somewhat similar. It's called Lower Mill Pond, and it has an alewife run into it as well. And we can see that almost immediately. By looking at the zooplankton, this is where if you did your zooplankton analysis, you'd say, whoa, something's happening. Well, in this case, it's intentional because they're encouraging the alewife run. But as a result, the water quality shifts very, very abruptly. So that's something that occurs even without the alewife. They're just an extreme example, and shows you that you would run from a body size in a really healthy lake of maybe 1.2, 1.5 millimeters, doesn't seem like much, but it goes down to 0.3 whenever the alewife are there. And all you have is rotifers, which is a completely different ecosystem. So the fish can have a big impact. So trying to keep that balance, if you have that choice, is really important. And I think it's very difficult to know by looking at a lake because you can't count the fish, and they're really the hardest things to assay in the lake, perhaps. But if you look at the zooplankton, the answer is right there because the fish are feeding on them, and it will tell you that. And there's been some really interesting papers done on that, and so we're trying to use that information to use that as a tool.
Well, it's 7:45. I do have one more question if you have time.
Sure.
And since you already said sure, it's kind of a big one. Not a big question, just maybe a big answer. And this is understanding that you're a man of science, not necessarily a public health expert, but I'll ask your opinion anyway. I think folks by now are used to the idea of avoiding the lake when there's a cyanobacteria bloom, and a certain number of colonies might produce a certain health risk. But the introduction of the concept of aerosolization and risks to air quality as well as water quality, and sometimes being having risks associated with cyanobacteria whether or not there's a big bloom present, can be a little, it can probably be a little scary to some folks. And we've talked a lot about the science tonight and kind of casually, but the implications are, potentially great. So do you have any thoughts or recommendations, and this question can be to either of you, on how regular lake users and folks that live on the lake and have lived on the lake for a long time, how people should be thinking about these health concerns and risks, and what kind of behaviors we should change, or just what we can be doing, or how we should be thinking about it, knowing what we're starting to learn about the potential health risks.
So yeah, you definitely kept the heavy artillery till the last. No, I think that's a really that's the question that motivates a lot of what we're doing, is because we don't have the answers to that question. We know that how much toxin is coming out of a lake, and we are looking at factors that can predict it. To some degree, it's related to how much is in the lake, as you might expect. So by reducing the toxin level in the lake by good practices, management, you try to keep that as low as possible. And yet what we're working on is we're trying to find what are the factors that will allow us to say how much is coming out of the lake without actually measuring it. And what are the conditions? So we're not quite there yet, but what we're also trying to do is develop a method, and this would be probably a laboratory method. We have what we call CLAMs. This is the compact lake aerosol monitors, and we've taken those at various places and used them. This is what our standard measuring device is. But we also know that we can bring samples into the lab and put them under laboratory conditions and actually ask the question, under these standardized conditions, how much aerosol is your lake producing? And in some ways, that's more satisfactory if you have a method that you can rely on, because if you go out to the lake, it may depend on the weather that day. It could depend on a lot of things. So we'd like to develop this method where if you have concerns about your lake because you feel it has lots of cyanobacteria, that you would be able to send us a sample, and we would then run an assay on it and give you the aerosolization potential of your lake. Is it one that produces lots or little, for example? And that may be just another extension of how we do monitoring. It's easier to do it with lake water, but air, because it's so transient, and it's hard to capture it. So, if we take the water and put it into the lab, then we set up our apparatus, we're able to measure how fast those toxins and cells come out of the water. So that's what we're working on now. We've got a project that we'll be doing at Lake Tahoe, where they have been concerned because of the number of cases of ALS in certain areas of the lake and nearby lakes, in fact, that have very high percentage, maybe 20% of the people living there have come down with ALS. So the question is, what's causing it? So this is a little different. It's sort of in response to the condition rather than avoiding getting the condition. They want to know what's the cause for this. And it's often that's an opportunity to learn something. With Legionnaire's disease, for example, the outbreak of Legionnaire's disease allowed us to learn what causes it, where does it come from? And we learned that, water that's aerosolized out of, that's evaporated essentially from air conditioning systems can harbor these particular viruses that cause Legionnaires. And so we think that it's the same, it's a possibility that we will learn something if we can go to places where there are high levels of ALS and maybe see correlations with what we're finding. We're also trying to identify what species of Picocyanobacteria come out of the lakes. And so we have submitted some samples to look at the DNA of those that are in the air that are coming out of the lakes that we're measuring, captured on filters, and to see if we can identify those in particular, because having identifiers associated with a particular disease might also tell us something. So, this is just the beginning of a pretty extensive problem, I think, that we're looking at. And how serious it is, we won't know till we answer it, So yeah. I'm hoping that it won't be as serious as it might seem to be, but we'll only know after we've done the research.
Yeah, it makes it that much more important for folks to get involved and start monitoring and start to at least get those answers as to what's in the lake right out of the gate.
Yeah. Our vision of lakes is unfortunately very biased because we are visual animals. And if we had developed extreme sense of smell that could detect microcystins in the air and such, we might know just from smelling it, like we can smell perfume, but you can't see the perfume molecules. And the same thing's happening with the lake water. We've got stuff coming out that we're just beginning to try to figure out how can we measure this and get past our vision.
Maybe we can get some cyano smelling pigs, like the truffle pigs.
Well, that, who knows? Don't underestimate what animals have evolved. That's a great idea.
Well, I've already kept you about 25 minutes past when I said I would, so I don't want to keep you any longer, but if you have any final comments, I'd be happy to hear them. And if you would like to share your email address, I can type it in the chat and folks could send you, folks could send you additional questions if that's okay.
It can be just jim.haney@unh.edu.
Okay.
So, yes, I'd be happy to hear from people. All I'd like to say is the last thing would be, again, to thank you for inviting me, but also simply say how much I admire your organization. I think it's exactly what we need more of, and promoting not only people interested in their lakes, but promoting science on the lakes and participating in that. So let us know where we can help, and that's all. If we have workshops, we'll let you know. Okay.
Good. Absolutely. We'll stay in touch and definitely keep working with you guys and keep the community connected. So thank you to both of you, and Jim in particular, thank you very much for your presentation this evening. And we will speak again.
Okay, great. Thank you, John.
All right. Thank you. Have a good night, everybody.
Night.
Thank you.